Now here we will be concentrating on the very first stage of meiosis 1, which is called prophase 1. Meiosis is a reductional division, and meiosis is quite an elaborate process because meiosis happens in two steps, that is meiosis 1 and meiosis 2. In meiosis 1, we have prophase 1, which is further divided into 5 substages called leptotene, zygotene, pachytene, diplotene, and diakinesis or terminalization. And what you notice here in these diagrams is that during the leptotene process, and they have colored the chromosomes in two different colors, the red and the blue chromosomes. The red and the blue together form pairs, and as you all are aware, pairs of chromosomes are called homologous chromosomes. The first thing is about leptotene. The first substage of prophase 1 is when the chromosomes become visible under the microscope. That means the DNA that is there, which has undergone duplication at the onset during the S phase, is beginning to undergo condensation. So because the DNA starts condensing, it starts appearing as rod-like structures under the microscope, which is nothing but the chromosomes. During zygotene, pairs of chromosomes get attracted to each other, and this process where chromosomes start pairing with each other is referred to as the synapsis. And I told you that two chromosomes come together, one is red and one is blue. The red chromosome is called, say we can call it as the paternal chromosome, and the blue one we can call it as the maternal chromosome. They are of two different origins, but the fact that they come together means that they are genetically identical. The coming together of pairs to form a bivalent or a tetrad. We can call a pair as a bivalent, or we can also call it as a tetrad, and see how they are coming close to each other. And this attraction between the chromosomes, the coming together, is called synapsis or tetrad. And when the chromosomes come together, see, each chromosome in a pair has two chromatids. So this is one member of the pair with two chromatids, and this is the other member of the corresponding member of the same pair having two chromatids. So this is a homologous pair which have undergone synapsis, and when they come together, there is a coming together, I told you, which is called synapsis. They are held together by a protein complex that is formed in between the two, and that protein complex is referred to as the synaptonemal complex. Now here you need to know, we would call the chromatids belonging to the same chromosome as sister chromatids. I would call these two as sister chromatids. However, this chromatid belonging to, say, this member of a pair, say I would call this member as A dash, and this chromatid that is belonging to the A member of this pair, I wouldn't call them as sister chromatids, I would call them as non-sister chromatids. Now why I want you to focus on the non-sister chromatids here is because the non-sister chromatids are going to take part in a process called crossing over. During this pachytene stage, they will overlap each other and segments will get cut off and mutually exchanged by a process referred to as crossing over. So you can see how they have overlapped here to form X-shaped structures, and wherever they overlap, the segments will get mutually exchanged. The ends of the non-sister chromatids will get severed, they will get cut off, and they will get mutually shuffled between the two non-sister chromatids. That process called crossing over is happening during the pachytene stage. Whereas what about diplotene? During diplotene, notice that the chromosomes have a tendency to move away from each other. Until now they were together as a synapsed homologous pair, but now there is a slight repulsion that is happening between them, and also the synaptonemal complex that is present between the chromosomes starts undergoing dissolution, and when they slightly move away from each other, you can see these X-shaped structures where they underwent crossing over, and these X-shaped structures where crossing over took place is referred to as the chiasma, and these chiasma become very clearly visible during the diplotene stage. And lastly, you can see during diakinesis, they move further away from each other, and another thing that happens during diakinesis is the nuclear envelope has started fragmenting, it has started disintegrating. We would call diakinesis as a transition into metaphase, because around the same time, it is not shown here in the image, but around the same time, the chromosomes have moved fairly apart from each other. They are not synapsed anymore, and also the fact that they align towards the equatorial plane of the cell as pairs, because now they have to step into the next phase of meiosis 1, that is metaphase 1. So in order to enter metaphase 1, they need to be aligned at the equator, and also by this time the spindle has been established by the centrioles which have migrated towards the opposite poles. So together all these 5 substages mark the largest or the longest phase in meiosis 1, which is called prophase 1.